Okay, so I'd like to finish by sharing with you both clinical and experimental data regarding, first, the role of biomarkers in predicting GVHD outcomes, and then what some of those biomarkers are teaching us about the biology. So what you see here are data from the Bone Marrow Transplant Clinical Trials Network, where we had 300 patients who had provided samples at the onset of GVHD. And the graph shows you the non-relapse mortality based on the presenting grade of GVHD for those 300 patients. And essentially those grades are not significantly different from each other, and this reproduces data from many studies that show that the clinical severity of diagnosis does not correlate with NRM and often does not guide treatment. When we looked at the biomarkers in those blood samples, we found that 3 of them, ST2, REG3A, and the TNF receptor type 1, could be combined into a biomarker signature with an algorithm that would weight each of the concentrations of the biomarkers and produce a predicted probability for each patient of non-relapse mortality. We could then rank order the probabilities and divide the patients into 3 separate groups, which we called the Ann Arbor staging system, since we had done that work in Ann Arbor, grades 1, 2, and 3. You now see again these same 300 patients according to their Ann Arbor stage. I'm not showing you the relapse mortality, but the probability did not predict relapse, and as a consequence, the overall survival from these 3 groups was significantly different. And these data were published 2 years ago with John Levine as the first author in Lancet Hematology. We used the non-relapse mortality as the endpoint for defining the algorithm, but as you would expect, the patients who had the low-risk group, who were in the low-risk group, would have the greatest response to treatment, particularly to steroid treatment. So here you see the complete response rate at day 28 in the 3 risk groups, about 75% for Ann Arbor 1, about 50% for Ann Arbor 2, and slightly less than 30% for Ann Arbor 3. What was most interesting in these biomarkers was the fact that they would, in the same group of 300 patients that were in the validation set, if a patient presented with Glucksberg 1 or modified Glucksberg 1 that's less than 50% of a rash, body surface area rash, what you see is 24% of the patients stayed at low risk, more than half of the patients became intermediate risk, and about 20% of the patients became high risk. And here you can see the non-relapse mortality for each of those patient groups. This same proportion of patients appeared also in the Glucksberg 2, so this is mild diarrhea as well as a more severe skin rash or liver disease. What you see is that we have approximately the same proportion of patients, slightly more than half are in the intermediate group, a quarter are in the low-risk group, and about 20% in the high-risk group. And perhaps most surprisingly, even those patients who presented with more than a liter of diarrhea and who would be considered overall grade 3 by this grading scale, a quarter of them were actually low risk, about half of them were intermediate risk, and a quarter remained high risk. So just if you would remember this going forward, according to this 3 biomarker signature, 3 biomarker algorithm, slightly more than half of the patients are still in the intermediate risk group, about a quarter are in the low-risk group, and about 20% are in the high-risk group. So this is the slide that you've seen before. We've heard about how the host antigen presenting cells are very important. We've got these donor T cells that are activating. The single best biomarker in all of the studies that have been performed, both by our groups and others, the single best biomarker is ST2. The vast majority of ST2 comes from the GI tract, and it comes from stromal cells and endothelium, some epithelium as well. So we believe that this is being released very early. ST2 is itself a sort of decoy receptor. It's a soluble receptor for IL-33. So what you see is that the cell becomes activated. IL-33 is an alarmin. So most of it is going to, when it actually gets into the bloodstream, is going to be bound to the soluble receptor. You've heard from Ernst Haller how the commensals are and their metabolites are very important and early on. We're now going to focus in this next little bit on one of the other biomarkers, which is REG3, which is produced by Paneth cells, as you heard from Ernst. It is stored in the mucus. It's a heavy molecule. It's stored in the mucus, and then it is driven in part by interleukin 22 that is secreted by these ILC3 cells. So REG3 and ST2 are going to wind up being major players in the next part of the talk. We knew from the algorithm at the onset of GVHD that many of the cases, for example, in that low-risk group, we could predict GI GVHD before there were symptoms. Those biomarkers were already in the bloodstream before we actually had GI symptoms. So we asked whether we could develop a new algorithm using the same three biomarkers, and in addition, we're going to put in the IL-2 receptor alpha, which is another single excellent biomarker that just fell out of the algorithm for the ONSET staging. So we're going to see whether we can predict non-relapse mortality and steroid-resistant GVHD prior to the onset of symptoms. So we're going to try to develop an algorithm at day +7 after transplant to predict severe GVHD and non-relapse mortality. For this, we're going to use samples from MAGIC, the Mount Sinai Acute GVHD International Consortium. The participating centers are shown here: Mount Sinai, the University of Michigan, Mayo Clinic, Ohio State, University of Pennsylvania, Emory, Regensburg, Hamburg, Dresden, Würzburg, and Bangkok. So this is a multi-continent, multi-center data set, which we deliberately have made so that we could try to develop an algorithm that would be applicable in any patient in any hospital. The data that I'm about to show you have now been published 2 weeks ago in JCI Insight. First author is Matthew Hartwell. So we use competing risk regression to model 6-month non-relapse mortality with relapse as the competing risk. The computer models compared all 13 possible combinations of algorithms of 4 biomarkers, of all the biomarkers, of all 4, for the most accurate prediction of 6-month NRM. We then used Monte Carlo cross-validation to test 75 different training sets that had been randomly generated to find the best combination of biomarkers. So there was one combination with all 4. The best combination of all 3 biomarkers was ST2, REG3 alpha, and IL-2 receptor alpha. The best combination of 2 biomarkers was ST2 and REG3. The single best biomarker was ST2. The best algorithm, which identified most accurately the largest number of high-risk patients, was the 2 biomarker combination of ST2 and REG3 alpha. So the final algorithm generates an estimated probability of non-relapse mortality for an individual patient, and then we needed to define thresholds that would divide the high-risk from the low-risk group. There were several such thresholds, and we chose the final one that gave us the largest number of high-risk patients with the maximum difference in non-relapse mortality between the high-risk and the low-risk groups. So remember, this is a single day 7 after transplant blood test. No patients have graft versus host disease yet. Here's the training set on the left and the test set on the right, about 600 patients and 309. You see the overall non-relapse mortality is 11 and 12% respectively. The algorithm gives us a difference of about 20% in terms of the overall the difference between the high-risk and the low-risk groups, and it's about 16% of the patients. That's essentially the best it's going to be because that's what the computer modeling is doing over and over again with logistic regression. So the first test is our test set. Here you see that there's actually a 23% difference, and it's approximately 17% of the patients. The validation set was those were all patients combined from the University of Michigan and Regensburg. The validation set is now from 9 centers in those 3 continents. It's an N of 358. There's an overall non-relapse mortality of 13%. We now have a 16% difference between high risk and low risk, and now it's about 20% of the patients who are identified as high risk. I'm not showing you the relapse data. They are published in the article, but there was no prediction of relapse, and therefore the overall survival is highly significantly different between these 2 groups. If we look at individual pre-transplant clinical risk factors, such as related donor or unrelated donor, what we see is that the algorithm works equally well. So in the related donor group on the left, you see that the algorithm gives you again a 20% difference between mortality, and 12% of the patients are identified as high risk. Whereas in the unrelated donor group, you have again approximately 20% differences in NRM, but now we've identified 21% of patients as high risk. So because we believe that the biomarkers are actually showing us subclinical GVHD damage, we're already incorporating into the algorithm some of these preclinical risk factors, and therefore there's high fidelity of the algorithm within each risk group. And this, we found similar results as are published, whether we looked at donor type, the age of the patient, the use of antithymocyte globulin as GVHD prophylaxis, or the intensity of the conditioning regimen. As you would expect, these algorithms do indeed predict specific GVHD outcomes. So this is acute GVHD-related mortality, which we define either as death from GVHD or death from infection while being treated for GVHD. So that's approximately 4-fold higher in the high-risk group. And here is steroid refractory GVHD. The difference is also approximately 3-fold higher in the high-risk group. Those were day 7 samples. We now asked whether that same algorithm, looking at 2 biomarkers, could give us the same risk stratification of patients at the time of onset of GVHD. So now we're going to essentially go back and do the Ann Arbor grades for these patients, but we have to look at samples that are taken at the onset of GVHD symptoms or at the onset of treatment. So in that was a large data set that I just showed you. It was about 1,300 patients. In 212 of those patients, we had onset samples. So we took them, we measured the biomarkers, and we used now the 2-biomarker algorithm and used the thresholds that would give us the same mortality strata in the Ann Arbor group, and this is what we found. We can indeed separate, using now 2 biomarkers, we can separate patients at the onset of GVHD into high, intermediate, and low risk for mortality. These are highly statistically different. In fact, the 2-biomarker algorithm is an improvement over the initial 3-biomarker algorithm because now almost 50% of the patients have been defined as low risk Ann Arbor 1, and about 27 or 28% are in the intermediate risk and the high risk. This is also published, but as you remember from our from the first slides, it was Ann Arbor 2 was the most with about 55%, and Ann Arbor 1 was then only 25%. We believe that the difference, the improvement here is largely due to the fact that between the first study in 2015 and this study, the commercially available kit that measures ST2 is now significantly more sensitive. We are now measuring picograms instead of nanograms, and that has given us a greater distinction, particularly in the patients between the low-risk group and the intermediate risk group. This algorithm also works after treatment. So Sunday, you will see a full presentation of these data, but we looked at about 400 patients 1 week after their treatment for GVHD from within this group. We're now using the same 2-biomarker algorithm. Here is our test cohort of 236. There's a 27% mortality. The algorithm divides the low-risk group into 12% NRM versus 50% for the high-risk group. We essentially see the same separation in our test, in our validation cohort. In data that I'm not showing you, we get very similar separations, whether patients have had a response to treatment on day 7. If they've had a response, it still turns out about half of those patients are at high risk and are going to have a high NRM. And even if they don't have a complete response by day 7, you can still see a significant number of those patients are actually at low risk. So now we have a single algorithm that can tell us about the future of GVHD outcomes before the onset of symptoms, at the onset of symptoms, and after treatment. So in the last couple minutes, let's talk a little bit about the biology. This is now early acute GVHD as opposed to very early. We see that these cells are damaging the intestinal stem cells that are known to be targets. Here's a Paneth cell that's divided. The ILC3 cells that produce the interleukin 22 are also decimated. There's a little bit of a paradox here. These are unpublished data now. If a patient has GVHD, we see that there are fewer, these are clinical data, there are fewer Paneth cells, there's less GVHD actually by staining, but there's a higher level in these same patients, there's a higher level of REG3 in the bloodstream. We went to a mouse model to understand this. Black is GVHD, white is not. And what you see is that in the animals without GVHD, there is this massive upregulation of REG3 throughout the villus, whereas if if the animals do have graft-versus-host disease, there's actually very little. That suggested that REG3 was important. So if you now do a transplant in a REG3 knockout animal versus a wild type, everything is the same, same donor, same recipient, except that the recipients can't make REG3. You see that there's a dramatic decrease in survival. You have even fewer Paneth cells if you use that as a semi-quantitative marker for histology. But the Paneth cells themselves are okay because they can reconstitute if there's no GVHD after the transplant. So it's not a Paneth cell defect per se. Here is H&E staining versus REG3. By the way, it's REG3 gamma in the mouse, it's REG3 alpha in the human. Those are the homologs. Here's a donor. You can see normal GVHD histology, no GVHD, normal histology, a little bit of REG3 staining. Here you can see the villus blunting of GVHD, and now that there's much less REG3 by immunohistochemistry. And if you give IL-22, which is the cytokine that is released by those ILC3 cells, you can see that there's restoration of the villus length, and now the entire villus length, essentially all of the epithelium is expressing REG3. And when you express REG3 in the epithelium and you essentially reestablish the barrier, the enterocyte barrier, in fact, so this is GVH, this is phosphate buffered saline. If you give IL-22, you're increasing REG3 in the GI tract and you're decreasing it in the serum. So the REG3 that we see leaking into the serum is essentially a surrogate marker for crypt damage. We then went on to say, does IL-22 require REG3 to work? So here is a wild-type animal with GVHD and getting phosphate buffered saline, and you can see just a little bit of REG3. If you give IL-22, you restore the villus length, and you see lots of REG3. Here's the knockout animal. The GVHD is even worse. There's no, as you would expect, there's no REG3 expression when you give IL-22. Here, these are the interferon gamma positive T cells, which are a marker of effector cells in the gut. You reduce them in the wild-type animals, but you don't change them in the softer colors, both for CD4 positive cells and CD8 positive cells. Both of them go down in the wild-type, but not in the knockout. And as a result, you can rescue these mice. In the wild-type, this has been shown before by Alan Hanash and Marcel Vandenbrink. You can rescue mice by giving them IL-22, but not if you can't make REG3. And that's true both in a minor antigen mismatch model of GVHD and a fully allo model. So REG3 is required for the beneficial effect of IL-22. So this opens up now, not only do we have biomarkers to look at early stages of GVHD, it starts to focus our attention on the innate immune system that's present in the GI epithelium and how we might be able to use it in terms of trying to restore health to these cells. So you can imagine now that IL-22 is inducing REG3, it's preserving what Paneth cells, enterocytes, and intestinal stem cells you already have. And this now takes us away from just immunosuppressive strategies to proactive, reconstitution strategies of the innate immune system in the GI tract. So we've developed a 2-biomarker algorithm that predicts NRM and lethal GVHD in over 1,000 patients. It predicts lethal GVHD early at onset of symptoms and 1 week after treatment. The algorithm predicts outcomes independently of pre-transplant risk factors or GVHD grade or response to treatment. It may be able to guide preemptive treatment, perhaps specifically aimed at the GI tract, and IL-22, which comes from damaged ILC3 cells during GVHD. Restoring it can help treat GVHD, but it requires the induction of REG3 gamma. Let me thank my collaborators, John Levine, Matt Hartwell, who did the predictive work, Hannah Major-Monfried, who's talking on Sunday about the response after treatment, Ernst Haller, Umut Uzbek, our statistician, Dongchang Zhao, who did the animal work, and our funding agencies. Thank you very much.